This is a video on the genetics of chronic fatigue syndrome. Chronic fatigue syndrome is a debilitating condition characterized mainly by novel, persistent tiredness that does not improve with rest. Its full clinical criteria has been hard to define due to its highly heterogeneous symptoms and frequent comorbidities. Diagnosis is currently based on the exclusion of other health conditions since we are yet to find a reliable diagnostic test or biomarker. It affects 0.2 to 0.4% of the UK population, is more common in women than men, and also presents a sizable economic burden. But what are its causes? Experts believe that chronic fatigue can be triggered by a combination of factors that include viral infections, bacterial infections, immune system problems, hormonal imbalances, physical or emotional trauma, and genetic factors. However, many of these factors remain inconclusive or of unknown significance. Chronic fatigue syndrome is polygenic, which means that a large number of alleles, each with a very small effect size, act cumulatively to predispose an individual. Several genealogical and twin studies provide evidence for a heritable predisposition. The study on the right estimates heritability as being 51%. This means that roughly half of the variation in disease liability can be attributed to additive genetic variation. So how can we identify the specific variants that cause susceptibility? A common method is to conduct a GWAS study, which scans whole genomes for single base changes called SNPs that are consistently different in cases compared to controls. However, to date, hardly any key variants have been confirmed and replicated in multiple studies. Nevertheless, many of the significant SNPs that are implicated, say, in the immune system, can now direct future research efforts towards which biological pathways and genes underpin chronic fatigue pathophysiology. Despite the strengths of these methods, the genetic factors that have been detected by GWAS studies do not collectively explain as much of the disease risk as we might expect from the heritability estimate, which, as we have seen, could be as high as 50%. This discrepancy may be explained by many low-frequency variants with small effects that potentially exist and are yet to be detected in a GWAS. Small sample sizes, insufficient statistical power, or misdiagnosed cases may well have hampered these studies. Another possibility is that epigenetic modifications account for a significant fraction of this missing heritability. Differential methylation patterns can cause long-term phenotypic changes in response to external stimuli such as stress and trauma. And such modifications have been found in chronic fatigue patients in association with pathological processes. Although it is difficult to prove causation, these modifications could be useful biomarkers for future clinical testing. Thank you for listening.